So welcome to the HPC, basically the using RAP for HPC users. So my name is Ted Ladaris. I'm a principal scientist and bioinformatics trainer at DNA Nexus, and my colleague is
Anastasia Sadlakova. I'm a senior community engagement scientist working at DNA Nexus, but focused on UKB RAP.
Yeah. So before we kind of get started, let's really kind of review things. And part of this was really kind of in our course of working with HPC users, we're just really kind of trying to map, their knowledge to what you need to do to actually run things on RAP. So definitely in terms of your needs, you want to be able to translate things so you can run scripts as you would on a on-prem HPC cluster. You want to be able to run multiple jobs in parallel, doing batch submissions. And ideally, you want to have a streamlined workflow. So I know a lot of you are very savvy on the command line. So, we'll reduce the minimal jumping between actually using the web user interface and the command line interface. So where possible, we're showing everything today on the command line. So that jumps into our learning objectives for today. So by the end of this session, you should be able to identify similarities and differences between HPC and cloud computing. Run any script, whether it be Bash, R, or Python, or other languages on the UKB RAP platform. Run single and distributed jobs. So just a quick note, if you have created a project on RAP, there is the ability to essentially refresh the data in your project so you don't have to redispense data. So under the settings tab in your project, you can click the check for updates button here, and that will basically refresh the data in your project. Okay, so this is a busy table. But I just want the main point of this is really to emphasize that a lot of what you know in HPC can be directly translated to cloud. So we've done our best to basically map the concepts onto kind of cloud over here. So this is where you're coming from, HPC. So there's a lot of things that basically kind of the concepts directly map, such as like when you are working with HPC, you SSH into the head node of the cluster. So that's your main point of interaction. But here with UKB RAP, your main point of interaction is the API server. Most of you have been coming from HPC, that you have submitted jobs with Portable Batch System or Slurm. With UKB RAP, that tool that you use to interact with the API server is the DX toolkit. One of the main differences, so of course, is that when you request a worker in HPC, that comes from basically a pool of machines in that private cluster. Whereas on UKB RAP, that's requested from a pool of machines in that AWS region. This is the critical difference, and this is why it's highlighted, is the idea of how do you get files in and out of the worker, and where do the files live? So in HPC, that's very much, there are lots of different kind of shared file systems that all nodes can see and access, whether that be a file system like Lustre or GPFS. On UKB RAP, the storage is based in what's called project storage, which is actually an Amazon S3 bucket. But the critical thing really is that, the implication is that because everything is in this kind of project storage, when you get to running files on a worker, you need to transfer the files that you need to run from project storage into the worker. And you need to transfer anything that you generate from the worker back into project storage. So one of the things we kind of do is we want to kind of make sure that we take your knowledge and we map it onto kind of the other the cloud computing framework. So let's talk about our mental models of computing, especially HPC, and see how it differs from cloud. So I think a lot of you are familiar with HPC, so I just wanted to highlight some of the key players and kind of show how they map onto cloud. Obviously, you're starting all of your jobs by from your own computer, and you're going to do some sort of SSH into the head node on HPC. And of course, the head node is the boss of everything on HPC. So like, if you're requesting something from the head node, then you'll get a HPC worker. And of course, you have temporary access to that. But again, this critical component with HPC is you've got that shared storage that not only the head node, but the HPC workers can see. So let's contrast that with cloud. Okay, so as you can see, like, hopefully everything is kind of mapping to the same kind of physical location. So you can see that how we're going to interact. And we'll be going over this process in just a little bit. But I just want to introduce key players here. So here is your computer, which you're going to be using to interact with the UKB RAP cloud. I'll just call that, for this sake of this, really the API server and cloud. You can kind of treat it as a single entity. And through that entity, you can get a temporary worker or allocation. But again, the main difference here is really that project storage. So you will need to get files to and from project storage to the worker, to and from the worker. Okay, so now we've established the key players. Let's talk about process. So like, you're going to start out on HPC by SSHing into the head node, and you'll basically get a shell on the head node. And then you'll submit a job. So when you submit a job, basically the head node is going to look for an allocation that kind of maps to your requirements. All of the files, because the worker has access to that shared storage, there's no file it's technically not a file transfer. It can be seen by the HPC worker, so you don't have to explicitly do that. But when you do the calculations on the worker, so here we've got a VCF file, and we're doing some sort of filtering on it. And so that basically, when we produce that basically appears back onto the head node. I know I'm simplifying things a little bit, because I know there is such a thing as scratch storage on an HPC worker, but we're just kind of simplifying things just so you can kind of map the concepts. So once you've gotten your, once the job is finished, you'll get a response from the head node that hopefully your job finished successfully. Okay, so let's look at the process on the research analysis platform, because it is slightly different. And again, part of that is really this project storage here. So on the research analysis platform, you're going to begin a command by doing a dx run command from your own computer. Again, this is done with that dx toolkit that we were talking about. There's going to be more instructions on kind of installing that. We will have a code repository available for everything that we demonstrate today. So when you do that dx run command, that results in a request to the platform. Platform looks at your request, and the platform looks at the worker pool and sends a request for the worker. So once you've got that, basically a software environment is installed here, and that you need to transfer the files over from project storage into the worker storage here. So once you have that, you can run your computation, produce your filtered file here. And then that needs to be this results file here, this filtered file, needs to be transferred back into the project storage. And hopefully your job finished, and you will get a response from the platform that your job has been finished. So like, and this is these learning challenges that we're talking about. These have really been kind of the experience that we have with interacting with customers, and who are new to cloud computing. So I just want to highlight a couple of things. So like we said, working with project storage is different compared to working with a shared file system on HPC. So there needs to be some sort of file transfer onto the worker of your input files. And then once you have them on the worker, part of the question is, how do you refer to those files on the worker to actually run them? And we'll get into that in a little bit. Let's talk about the second challenge. So once we've finished our computations, we need to get our files from the worker storage back into project storage. So hopefully this isn't too hypnotizing, but I think one of the challenges with working with cloud computing is that there's a kind of nestedness to this. And we'll be talking about that in a little, or Anastasia will be talking about that in a little bit. But hopefully like this helps, like, orient you and kind of helping you understand how your own mental model of HPC maps to cloud computing. And Anastasia will now talk about the next section.
So Ted was talking about mental models of cloud computing, and I will be walking you through how you can run apps in the cloud, how you can actually use these mental models in order to do some practical work in the cloud. But before we'll start that, I would like just a bit introduce our platform for those of you who was not using it before. So we are a highly secure platform, and we're highly configurable. So you can run either single scripts or fully automated production level workflows. And the data transfer is very efficient. So instead of waiting for the data to be downloaded on your HPC for days or weeks, it usually takes about a few hours. And also, instances are configured via apps, and you can configure, there are different ways how you can configure it. And you can use, for example, Docker, and we'll be talking about that in a moment. And also, because we're working on the top of AWS, you can access the wells of AWS system. So here, I just want to show something that Ted will be go more into details, and we'll be actually also mentioning it, is that when you have a project storage on UKB RAP, you're using Amazon S3, and then you can move your files with to the worker instance to the Amazon EC2. And you can use for that dxFUSE, and Ted will be showing it to you in the end of this webinar. So this webinar actually is made to map for you what you are used to do and what you will do on the cloud, particularly on UKB RAP. So here we created a table when you can with common commands that you can use either in a portable batch system or Slurm, and the one that we will be using on UKB RAP. So we will be using dxRun command to run the job. We can also monitor our jobs either by find jobs or watch. And if we want to terminate our job for whatever reason, we can use dxTerminate. And here you can see the corresponding command that you will use on your HPC. So this section is called running your apps. And this is because apps is the heart of our platform. So here you can see some of the subset of apps that we have. And apps is in blue are general purpose apps or all purpose apps. So what app can do for you on the UKB RAP is at least request a worker, EC2 worker for you based on what you configured in your configuration, and also establish data transfer from the project to the worker and back. And for our example for today, we'll be using Swiss Army Knife, which is a generic app that is used for some common bioinformatics operations. So here you can see different tools that it has. It also has some general purpose tools like R, Python, and standard Linux commands. And why it uses standard Linux commands is because all of our instances are based on Ubuntu. So that's why we can use Linux commands there, and all of the bioinformatics tools that are running on Linux. And here you can see the documentation page for Swiss Army knife. The only thing is that you need to be logged into RAP in order to access this. So why are we using Swiss Army knife? And this is let's say it's something like new concept for if you're coming from HPC. So this is a generic app that, as I said, where we can do the common operations. It has some pre-installed tools. And what is very important is that in case the tools are not installed there, you can also use Docker, either with image, when you are just linking the image link to the Docker Hub, or image file for the Docker image file in your project. So, and also Swiss Army knife has very rich documentation when it shows you some of the examples how you can use different bioinformatics tools with files. So there's a lot of nice examples, and I really advise you to go through the documentation before you start with Swiss Army knife. So what are the practical approaches to work on RAP? So as Ted mentioned, we'll be using dxFUSE for the single jobs, either or command line, and this part will be devoted to that, or in a shell script. And then Ted will show you how you can use dxRun for batch processing, or how you can use dxFUSE. The thing that we will not be covering today is dx generate batch, but we will here is a link to documentation, and the slides will be available to you afterwards, so you can self-study that. Okay, so now we were talking a little bit about Swiss Army knife. Let's again map what you know from your HPC to what we have on UKB RAP. So if you have pre-installed tools, you will list modules that are installed on your HPC, for example, using module list. And because here we will be using VCF tools, so all of our examples, almost all except for batch processing, will be on VCF tools. So if it's installed, you can use tool in the script like module load VCF tools. With UKB RAP, when we are using Swiss Army knife, you will look into documentation to check whether the tool is installed in Swiss Army knife. And if it's installed, you will just run dxRun app Swiss Army knife, and we'll be digging much more into that in a few slides. And we have an ICMD parameter, which is a command line parameter, and there you will be just using the tool without any installation there. So this was about pre-installed tools. What about tools that are not installed? You can use them with Docker image. So usually on HPC, you will not use Docker, but you will use Singularity. So here, for example, you are pulling Singularity, using Singularity, the Docker, which contains VCF tools. And then you can execute commands like using this Docker image. On a UKB RAP, when using Swiss Army knife, you can just specify a link to the image on Docker Hub. So like that, using image parameter, and here we're specifying the image link, or this part remains the same, but here we're having image file in case we're downloading the image file on the project. And why we could use this option is because there is a download limit from Docker Hub. So that's why we recommend you to download this image and then use it from your project storage. So when we were talking about the cloud computing and the mental models, is the one thing that you really need to realize is thinking about 2 steps ahead. So first, where your data are, how you can transfer it to the worker, and how you can transfer it back to the project. So here, in our example, we have our VCF file. As you can see, the EID is not the real EID due to the fact that we could not share with you EID's numbers. And we will be transferring the data into the worker. Then we'll be running computation, and we will generate result files. And this is command that we'll be running, but don't worry if you don't understand. We'll be really going into the details just a few slides later. And once we have our filtered VCF file, our output file, we would like to save it into the project. And the challenge is visualizing where the files are in the worker, and we'll be looking into that. And also, what is benefits when you're not just programming it by yourself, but using Swiss Army knife, is that outputs are automatically uploaded to the project storage by Swiss Army knife. Okay, so these were some basic introductions into how we would use our Swiss Army knife. And now let's look into our example. So the code for this webinar is available on our repository, UKB RAP. Here is the link, but you can go on our repository right now and you can find the code. It's already there. So when we run Swiss Army knife, here just to map UI and command line interface, as Tess said, most of you are probably technically savvy, so we will not be using UI here for you, just in order to map here. So here is a print screen for Swiss Army knife. Here are some input files, which we will be using as IIN, which is like interactive. That's why it's I in the beginning for the app. So that's why it's kind of like, it seems strange when you see it -IIN with only one dash here. And that's why we also see I here, ICMD. It's a command line where we will actually be writing the command that we want to run on the worker. So here is the in file, which is here, and here is the command parameter, which is here. And the files that are that we're specifying here in our IIN parameter. So here's our VCF, our gzipped VCF. It's transferred into working directory of the worker by default. So that means that we do not, this file path will not be existing, so we can just call it by name without specifying file path. But we will be showing it to you in a minute. So our example, our ICMD part. So remember the ICMD, the command part that we'll be doing here for today is filter by genotype, death, and quality. So let's go line by line so we know that we understand the command, and then we can focus on how we can use this command specifically on UKB RAP. So we'll be using WCF tools for that, the tools that are already installed on Swiss Army knife. We'll be using compressed gzipped VCF that we specified here. Also, we'll be filtering by genotype quality and genotype depth and specific values. We will be creating output file as an uncompressed VCF. And here is the prefix of the file name that we will be creating. Okay, so this was our command that will be actually running. Let's look how it will be all working when we will be submitting it via Swiss Army knife. So this is a bit busy slide, but let's go line by line. So dx run, as we may remember, is the dx toolkit command, how you can run up, and the app name is app Swiss Army knife. How do I know that it's app Swiss Army knife? So now I know it by heart, but before I know it, I was just looking into documentation. And in case you are not sure, just look into documentation on UKB RAP and you will see the dx run example command, and then you will see the app name. Then we have one input file, bulk / something 1, 2, 3, 4, 5, 6, 7 PCFGZ. And as we were saying, this file is transferred into working directory by default. Then is our, so that's why in our ICMD parameter, we are referring to it as just 1, 2, 3, 4, 5, 6, 7 without this file path. Okay, so this is the biggest difference. Yeah. And so the ICMD parameters here is the one that we were talking about a few minutes before, a few seconds before, the VCF tools filtering and outputting. And -y is that the DX toolkit will asking us if the settings are okay, if the DX toolkit can run the command, and we would like to answer yes on that. So that's why. So in this example, what is kind of like annoying is that in case you will be running it on multiple files, you don't want to retype it again, like again and again, different file names. It's just not convenient. So what you can do is you can use Swiss Army knife special keywords. And we have two special keywords that we can use. It's $in underscore name. It's a file name of the first input file, or $in underscore prefix is the prefix of the first input file. And this can be helpful for naming, so you don't need to hard type it. And the interesting thing is that you can also, in case you have more input files, and I will be showing to you an example of that, you can iterate and index them as you would index array in Bash. So let's look at our example as we have it, but upgrade it. So what we have it here hard-coded. We have hard-coded our input file, which we need to show the exact input file, but we also have hard-coded the same input file name here as a specifying gzip2ecf and also specifying output prefix. So what can we do about it? We can just use, instead of the exact file name, in name for the gzip2ecf, and in prefix by combining it with filtered for our output prefix naming. Okay, so I'll just flip back and forth so you can see the differences. So here are the hard-coded name, and here are the special keywords. Also, if we would like to upgrade a little bit, let's say that we have a WCF file and we would like to filter it by the list of the positions. So now we have two inputs file, the WCF file itself and the text file with the positions. Again, hard-coded WCF files here, hard-coded WCF file, TXT file for the positions. So what we can use special keywords and index them. So here you can see that this is the first, the WCF file is first, that's why we use index 0. TXT file is second, that's why we use index 1. Again, I will flip back and forth so you can see. Here's hard-coded, here's with the keywords, okay? And the last, like, even more modification to that, the files will be transferred after the Swiss Army knife is finished into the root folder of project directory. But in case you want it to be transferred somewhere else, you can specify it by using -- destination parameter and specifying their project and your folder. And if folder does not exist, the Swiss Army knife, or like the platform, will create it for you. So this was, we submitted our job, we created it. So what will we get? We'll get job ID, and we can use this job ID for our to watch it. So here we're using dxWatch, and here you can see that we can see the progress here. And once the job is finished, hopefully successfully, the file is created in the project. So here you can see that we have our WCF file here. So what we just learned is that we can run Swiss Army knife using dx-run app Swiss Army knife, and you can search for app Swiss Army knife in documentation. The inputs in the command line are specified with -i, so that's why it could be like a little bit strange that you have ICMD and IIN. So it's important that you know that it's -IIN and not --IN. And when files are transferred, they are transferred into working directory of the worker. So there is no need to have a file path, the full file path as you have in a project. And any outputs of the worker are saved into the project storage in the root directory. And if you want to change it, you need to use a destination parameter. So next part that that I will be showing to you is how we can now use so before we're using command line interface, but what if we'll be using it in a script? And here again, we'll go back to HPC and map it there. So our goal is the same, because this is not main focus of our webinar. But what is the main focus is to map as much as we can UKB RAP to HPC. So we're again filtering using dxFUSE. And here's a little bit different. What we are doing is we're creating my_script_run.sh, and inside we're putting our command. So far, this part seems the same. We have a command to run, but what we added here is we have an instance request for resources. So in the documentation, you can find the default instance, but if you would like to configure it, or if you would like it to be explicit, you can add --instance--type parameter, and then you can have the instance name. So where will you get instance name? That's why we have a list of currently available instances on UKB RAP. You can click on that link and you will see the different names that you can fill this in. So, okay, this was our UKB RAP. So we're basically putting our dxRun command into the script. How it's look with a portable, on your HPC if you're using portable batch system. So here is our instance type. It's here with a parameter, and here it's specified in the PBS. And this could not be 100% mapped with your HPC. So that's why we really encourage you to look into this link, the available instances, and map and find the ideal for your particular job. And the second comparison that we have here is our ICMD part. So if we're running here with dxFUSE, this is how we specify it here on UKB RAP with ICMD. And here's how we specify it in HPC in our script. Okay, so here's again mapping to that. So what we just learned is that we can just place our dxRun command into the shell script and run it on your own machine to start a job. So you run the script on your own machine, but by running dxRun, if you remember the first slides from Ted's part, is that you're actually creating an API request to the UK Biobank RAP platform. And this will be actually very useful when we start batch analysis, and Ted will talk about will walk you through that part.
Okay, so we talked about some of the fundamental skills about running things with dxRun. Now we're going to kind of take it up a notch. So not only are we going to run it, we're going to run dxRun on multiple files, which is probably the case that most of you will be doing on RAP. So our goal with this section, so we wanted to make this kind of relevant to, as many people as possible. So our example in this section is going to be really working with Plink. And if you have worked with Plink before, the default file format that you need is a triplet of files. So that's that bed, BIM, and FAM files. And our goal is that we've got, and this is aggregated data. So this is not, these are not EIDs. This is just the field ID. So we're going to basically be processing each chromosome triplet on a single worker. So you can see that this one has C1. So that corresponds to chromosome 1. Similarly, C2 is chromosome 2, and so on. So this is a little confusing, but we did want to generalize this approach as much as possible. And again, this code is going to be available in the repository. We've tried to make the documentation as clear as possible. So there's going to be two kinds of scripts. So we talked about kind of running our scripts on our worker. So all of the scripts that we use for batch processing are going to begin with batch. So this is going to be a batch run.ssh or .sh. Our command that we actually want to run, and again, we're doing this because we want to generalize, being able to run all kinds of scripts on our workers, is that we have a script called plink_script.sh. So this is going to contain the Plink commands that we want to run on our worker. This is going to be stored within project storage, and there's much more details about how to set this up in your project, but essentially you're going to put this script in a folder called scripts in your project, and then you can basically run this. So before we kind of, again, we're just really trying to map knowledge, our previous knowledge. And let's talk about how we would basically do batch processing on HPC and compare it to how we do it on the cloud. So really on HPC, our main mechanism for processing multiple files is this idea of a job array ID. We'll show the code in a second, but the idea is that we say we want 22 jobs, so we want to process 22 chromosomes. And then we can basically use those job array IDs to process one file triplet per worker. And this is all within our submission script. Once that's ready, we can submit our job to our head node. So cloud, the recipe is slightly different. So we're going to have basically our shell script here. We'll list our files, but so we'll use a for loop within our shell script to basically spawn a dx run command for each triplet. And then within that dx run command, we'll transfer the relevant files and then our plink script, shell script, and then we'll run the plink script, shell script, that is very hard to say quickly, on our worker. So that was like the basic kind of pseudocode. So let's actually compare the different kinds of code. So really what I want you to focus on here is really that with PBS, you have this job array that you're specifying. In this example, we're only processing the first 5 chromosomes. And you basically use that, there's a placeholder variable for that array ID that you're going to use to process the file. With UKB RAP, instead of using that job array ID, what we actually use is a for loop here. And within our for loop, basically, we're going to put our Swiss Army knife command. And then we are going to use that, basically in our for loop, we have that placeholder variable. We'll use that to run our files. So for example, this is going to range from 1 to 5. So the command that's actually going to be run on the worker is bash plink script .sh, and then is going to use like the number of the chromosome as an input. So we'll dive into what exactly that's doing on the worker in just a second. But here, really, again, like execution wise, on HPC, we need to execute this on the head node. With OnRAP, we can actually run this batch command anywhere that we have shell access to, and it will basically do our submissions using those multiple dx run commands. Let's see. So I think I already went over that. Okay, so let's talk about what the plink script is going to contain. So this is a little bit of a tricky bit. You'll notice that in our script, basically instead of I here, we have a 1 here. So that is it means it's taking the first argument that of the script. So this is going to be run on the individual workers again. So what this is going to be doing, if you know how plink works, is that the way you specify a triplet is with this B file parameter. And here we're basically, this is kind of the prefix of the files in our directory. And this is going to basically range from 1 to 22 here. So it'll be, for example, UKB 24418_C1, and then with this suffix here. So that's how we're using that kind of first argument. And then we'll recycle that in our output. So we'll generate a file called CHR. For example, for the first one, that would be chr1.results. So that's going to be the prefix for our results files. If you've done this kind of filtering and analysis, you'll know that basically there's going to be multiple files generated. So this is going to be kind of the prefix, and there'll be various files that'll be generated. So like, for example, the bed file that's generated from this will be chr1.results.bed. So let's talk about, what happens when we actually run that batch script from our computer. So on our own machine, we're going to be executing that. This could be either run with sh or bash, depending on how you like it. What that's going to do is it's going to basically send a series of dxrun commands to UKB RAP. UKB RAP is going to process them and say, okay, so for our first iteration, we're going to work only with the chromosome 1 files. And so it's going to transfer our Plink script and those relevant files over. And then we're going to process them on that worker. So this is going to produce all of the chromosome 1 files. Similarly, and I don't have all 22 because that would just be a way too busy slide, but here's the second chromosome. So basically the Plink script and the second chromosome files are going to be transferred over into worker 2. And then it's going to process that. It's going to produce our results in each of them. And I know there's going to be multiple files generated here, but all of these, when the worker is finished, it's going to be transferred back into storage. So when we do that, basically this is how, if everything works well, this is just for kind of the first 3 iterations. You can see that we have generated our script here, our 3 different kind of batch submissions. So this one is for chromosome 1, chromosome 2, chromosome 3. And you can see that thankfully they finished. And that you can see that we actually store our results in this results folder here. So this is a little bit of a whirlwind tour, but really I just want you to focus on the fact that you can actually execute shell scripts on the worker by just including them as an input file in your dxrun submission. And then when you actually have those shell scripts on your worker, you can actually specify in your ICMD command input. So that command line input, you can use bash and the name of the shell script. We can use a combination of shell scripting and for loops in our batch submission to spawn 1 worker per file using dxrun. And again, thinking about the placeholder variables and how they're critically important to specify which files to transfer to the worker and specify the file output name. So what we showed you is kind of the basic recipe for batch processing. So there is an alternate way to access files, and that is with the dxFUSE file system. So this is going to enable file streaming. So you don't have to explicitly transfer the files over. There's a way to kind of specify this. Again, part of this seems like, we're going at a bit of a plodding pace, but really this is to kind of contain the flow of information. So we're just trying to show you one piece of information at a time. So it's a little more manageable. So there is a streaming system called dxFUSE. And this is a shared file system for the workers. We were debating just showing everything with dxFUSE, but this is currently in beta. So we want to show some firmly established path on how to kind of process files. So like in this case, we don't have to actually explicitly specify our files as inputs. We can actually directly access them from project storage using this dxFUSE file system. The way we do this is that in our file paths, we can pre-append a /mmt/project to the path of the file in project storage. So here's an example. So instead of having /raw_data/plink, we would put a /mnt/project/raw data. And there's going to be in the code example, in the code repository, there is an example that works. So definitely, hopefully you can use that, you can alter that for your own purposes. Like I said, dxFUSE is currently in beta state. So really we encourage you only to use it for read-only files. There is write file support using dxFUSE, but the support is currently limited. And it's, so if you really want to make sure that files get back into project storage, you want to basically use kind of the other methods that we talked about. So let's just look at how we would alter our script. So this is, again, the script will be available here. So the main thing that we're going to transfer over in our dxRun command is just our plink script. So it's called in here called plink _script _dxFUSE.sh. And here, basically in our plink script, we can, in that v file parameter, we can put the full path with that placeholder variable to basically specify the triplet. So here's an example of that, and you can see that it's executed on the worker. So just want to highlight here. So this is that parameter where we put kind of the entire path with that /mmt/project. And that is going to run, that's basically going to run plink. It'll stream the files from project storage onto the worker. And, the way this is set up, all of the jobs are numbered. So you can see that basically it just takes a little bit and then the jobs will finish. And there's hopefully a happy ending and all of the files will be generated into our project. So what did we just learn? So using this /mmt/project with the file paths lets us basically stream files from project storage into the worker without us explicitly downloading. Just again, to emphasize that this method works best for reading files. Currently, there is limited write support for dxFUSE. So that hopefully kind of completes the picture. But again, we always want you to expand your skills and knowledge. So there are there's multiple ways to do that. So in terms of things we want to do, dxGenerate, there is an alternate method called dxGenerate batch inputs, and that uses regular expressions to specify file inputs, and that generates a tab separated file that can be run with this special version of dxRun. You can learn more dx toolkit commands, and again, this is the key to becoming a power user on RAP, is really understanding working with metadata and using the dx find data command to select a lot of files on the platform to iterate over is like a really important skill. If you don't like the apps in Swiss Army Knife, you can build your own apps. This is a link to our webinar about that. There was a question about WDL and Nextflow. There is our WDL Docker webinar as well. And then this is if you want more information about working with Swiss Army Knife, this is information on how you use Docker images with Swiss Army Knife. You do need to have a RAP login to access them. Just want to talk about, when you're signing into the platform at ukbiobank.dnanexus.com, there are some useful links here under help and in the main page. So these are links to our support, documentation, and community. And please use community. We know that everyone has lots of questions, but asking them there will help everybody. Anastasia, would you like to wrap things up?
Yeah, here's just link to the quick start on our documentation, some previous webinars, community, we already listed that. And there are 2 GitHub repositories. One is with materials for the webinars, and another is with some example notebooks. And I think that, upcoming webinar, there was a question about if you can use R. Well, you can use R, and actually our next webinar is about how you can use R on UKB RAP, and it will be on June 19th. So definitely, in case you would like to use R on our platform, we definitely advise you to register on that webinar. And on the September, because we'll have a summer break, we'll have a section or webinar specifically focused on Docker. And then you have linked to all of the webinar recordings, because I think it's more than 10 to current moment.
Yep. And this is just a quick link. So if you want a QR code, definitely here. So let's just take a look at Q&A in that limited 1 minute of time we have. So just again, slides are going to be made available. There are lots of questions, especially, yes, so like as Anastasia mentioned, the question about running R scripts on RAP, we will definitely cover that.
Maybe I have one like that, or a few that I would like to pinpoint. There was a question about the platform support Nextflow or WDL workflows. So for WDL, we have a DX compiler tool, and in case you are interested in that, we had a 2-section webinar on that, 2 parts. And for the Nextflow, you can run it on, for example, TTYD or Cloud Workstation. And in case you are interested in that, please drop us a line. So this will help us to prioritize coming webinars. Do you have some favorite of yours, Ted? The last question that you would like to?
No, I mean, they're all great questions.
That's true. Thank you for being so active. This is really make us very happy as a presenters.
Yeah. And please, again, if you have any questions, please reach out via community. Again, many people will probably have your same questions. So you are not only helping yourself, but you're helping everyone else in community. So thank you.